This is the Business of Biotech. I'm Matt Pillar, and sitting across the desk from me is Geoff MacKay, founder and CEO at AVROBIO. Geoff, welcome to the show.
Thank you. Pleasure to be here.
Oh, it's great to have you. So I'm going to share a little bit about you with our audience. Geoff began his life sciences career as VP of Immunology and Transplantation at Novartis before jumping into the CEO's chair at Organogenesis back in 2003. During his 11 years there, the company treated 1 million patients with living cell therapies, which is an accomplishment that fueled Geoff's fire to refine cell therapy manufacturing processes for at-scale production. Geoff went on to found eGenesis Bio and served as chairman of the board of MassBio and the Alliance for Regenerative Medicine before launching his current company, AVROBIO, in the spring of 2015. Now, Geoff, we're going to talk about your mission to create scalable cell therapies. But first, we need to get to know you a little bit better because your background is not typical of a biotech entrepreneur. When you first came out of college, you were on what looks to be a completely different and unrelated career path for a few years before you joined that transplantation and tissue engineering team at Novartis. So tell us about that and why you made that sharp turn into life sciences.
Sure. So when I finished my graduate studies in management at McGill in Montreal, Canada, where I'm from, I really didn't know what direction I was going to go in. And back in the 1990s, Nortel was probably the coolest high-tech company in Canada and really attractive for young people. So I was really happy to get a job in marketing at Nortel. However, after about a year or two, I realized that telecommunications was probably not my cup of tea and not something I wanted to do for my life. So I actually went to work as a consultant at AC Nielsen. And that was fabulous because I found myself as a consultant in the C-suite of, a dozen different companies. But my two main clients were Kraft General Foods and Merck. And so half my week was focused on the cheese division of Kraft General Foods and half in the high science realm of Merck. And it was a fascinating contrast. And I have to say that, the people at Kraft General Foods were brilliant, smart, creative, that they could predict based on an end dial display what the market share across the country would be 2 months later. So really sophisticated, analytical. However, it was cheese. It was slices, Velveeta, cheese loaf, cream cheese. And so I eventually decided that, I didn't really want my career ambition to be head of the cheese division of Kraft General Foods because the other half of the week I could just see the vision of Merck and the mission to really change and save the lives of just thousands and millions of people. And so although the culture was a little slower, a little more stodgy, the ultimate gratification of actually being in the life science industry was profound. And so as a result, I left AC Nielsen and got a job at Sandoz, which eventually merged with Ciba-Geigy and became Novartis. And that was also a wonderful early experience because Novartis is such a great school. They invest in their people. And I actually did 11 jobs in the Novartis organization in 11 years, and beginning in Canada, in the US affiliate, and in the head office in Basel, Switzerland, culminating primarily in, as you said, as VP of Transplant Immunology. Yeah. And then, I think part of Novartis was this early dabbling in this new field of cell and gene therapy and xenotransplantation. And Novartis got in big in the 1990s, but then I think realized that it was too soon and pulled out, but it gave me that exposure. And because of that, I just decided that I would take a chance at this new exciting field of medicine and went to build out Organogenesis for the next stage of my career, which was the next 11 years.
Yeah. So, it's sort of an abbreviated history. I'm curious, just a follow-up question on sort of the trajectory of your career. At some point, you went from consulting and two great big giant industries, decided you liked one sort of better than the other. One, I mean, do you think I'll make it a two-part question.
Mhm.
One, do you think you may have found the life sciences and pursued that had it not been for that consulting gig at Nielsen? And two, at one point after you made that hard turn into the life sciences, did you actively pursue entrepreneurship and leadership, management of these companies? It's one thing to say, hey, I'm going to, focus my career in the life sciences. It's quite another to risk not once but twice, everything to found and lead a startup.
So I've wondered that first question myself, and to be honest, I don't think I would have found the pharmaceutical industry were it not for AC Nielsen and just being plopped down inside these organizations. So I'm extremely grateful for that opportunity. Because I think at that point my education was in management, my interests were broad, and I really hadn't zeroed in on it. So I think that in that sense I'm extremely grateful.
Yeah. So let's and then there again, you went from taking that leap into this industry to founding two companies, and now you're working in earnest to do things that have never been done before, the at scale manufacturing of cell and gene therapies. So let's talk about that. What, in your experience now having led a couple of companies in the space, what's been the historical challenge to scale?
So a lot of new technologies have challenge with scale. If you go back to monoclonal antibodies, at first the industry couldn't make them, and eventually those challenges were conquered with large scale bioreactors. I think that the point in time that the cell and gene therapy industry is at right now is that there's some really cool science. We're able to produce enough for clinical grade material. And what's emerging are potential, really a potential revolution in healthcare, potential one and done curative therapies. But one of the elephants in the room is, how do you take this Petri dish science and to be able to mass produce it and turn it into pharmaceutical grade medicine? Mm-hmm. And there's different types of cell and gene therapy. Our particular focus is lentiviral gene therapy, where you actually transduce the patient's own stem cells to deliver the healthy gene and then reinfuse back. And the manufacturing challenges there are formidable. And I think that now that clinical validation is really here, our thoughts at Avro and the industry in general is really focused on how do you get this type of technology anywhere around the world at scale that's cost effective and convenient. So based on my 11 years at Organogenesis, where we faced a similar challenge in the first generation of regenerative medicine products, which were non-gene modified cell therapies, we really wanted to do the same thing again to gene therapies, which is to take each component of the process, break it down, figure out how to optimize it, how to scale it. And a lot of times this means applying innovation, meaning how do we automate manufacturing? How do we, incorporate things that used to be done manually into a roboticized process such that, we can really, really increase scale, get human intervention out of the process. Us humans are dirty animals, and the less we touch the process, the higher the quality. And to be able to do it in a way that is reproducible such that the same drug product comes out every time. Yeah. And, I think that that's where we are as an industry right now. But whether you're talking about monoclonal antibodies, small molecules, or widgets, the same thought process happens. Is once you have a validated process, you ask a different question, is how do we mass produce it at scale that is cost effective, that has a consistent set of quality parameters. And so this is a key focus, of course, at Avro, but it's also now becoming a focus of the industry in general.
What's the mentality there around, so your mentality, I should say, around decision making? Like, you could look at it and go, okay, at scale, the tech is there. It's time because the tech is there. Or you can look at it and you can go, The tech isn't there, but we're going to go make the tech.
Mm-hmm.
Where would you kind of, and I know those are two extremes, right?
Yeah.
But so maybe there's somewhere in the middle, but kind of frame that up for us. Your mindset around, I'm gonna go all in on this because it's time, or I'm gonna go ahead and make it time.
I think we're going to go all in on it because it's time. And if you go back to 2015, I really wanted to form a cell and gene therapy company, but the catalyst to do so was the fact that clinical validation was popping after perhaps 2 decades of false starts in the gene therapy world. Finally, it really seemed as though gene therapy was becoming ready for prime time. And you could look at places like UPenn were curing cancer with ex vivo gene therapy. I mean, just phenomenal, which the CAR T world. And companies like Bluebird Bio down the street were showing curative effects with gene therapy, ex vivo lenti gene therapy. Companies like Spark were coming out with really impressive data with AAV gene therapy. And so it just seemed as though after decades of academic work, false starts, industry and venture capital jumping in, pulling out, the field sort of going into the dark ages, that finally there was this consistent pattern of this is working. And some of the early kinks related to safety had largely been worked out. So then, although it was working, it was absolutely a know-how intensive field. That this is a tough slog to be able to take this petri dish science and turn it into a medicine. And that's where I just decided that myself and 2 other founders had those skill sets. I had been running the largest cell therapy company in the world for a decade plus, and really the only model that had actually scaled, the only company that mass produced living cell therapy. So on the business side, I came with that heritage. The other founder, Chris Mason, was chair of regenerative medicine at UCL, which is one of the powerhouse schools in gene therapy, probably the number 1 school in the world in London. And so he had a lot of the translational science background. Mm-hmm. And our third founder, Kim Warren, was head of process development at Lonza, which at that time, all roads led to Lonza. No one had built out manufacturing, and Lonza was pretty much the only manufacturer that scaled up cell and gene therapy. So Kim and her team of 50 had scaled up almost every type of cell or gene therapy that there was. So manufacturing science and business, we just felt that now that validation was here, we could build a company around it. And so with that nucleus of skill, we just spent the first year turning over every rock in academia, going around the world looking for unique, differentiated assets that were not science projects. We wanted pre-validated, in essentially preclinical data that showed that these gene therapies were ready to be translated into the clinic, because that was really the skill set that we thought we could add. So to take the baton from academics and get this great science into the clinic where we could see it work in real human trials.
Mm-hmm. Okay. And then to, as I understand it, to take, to move toward off the shelf gene therapies, Avro decided to pursue an off the shelf manufacturing strategy, and that didn't work. I'm oversimplifying it. I'm going to let you tell us the details, but it didn't work for you, right?
Well, yeah. So I think the premise is that if this was easy, others would do it, right? And
sure.
There's something inherently attractive about automating manufacturing. Just like in every other sector, every industry, eventually you get there. I think that the point in time that we're at with gene therapy is that no particular model fits perfectly. So we learn from small molecule approaches, we learn from biologic approaches, and we learn from bone marrow transplant approaches. And there's particularly effective automated manufacturing for bone marrow transplant. After decades, they've really got to the place where there are fully automated pods that can mass produce for bone marrow transplant. And so as a starting point, we took that technology, those robots, but clearly it's not off the shelf. And we then had to spend years optimizing them and essentially refining the algorithms of these automated pods such that they can produce, cryopreserved gene therapy as a final drug product with a consistency that is really impressive. And so where we are now is having those automated pods situated in manufacturing facilities in California, in Houston, in Melbourne, Australia, and in a few short months in Europe. If the operator presses the go button, the same drug product comes out, whether that go button is pressed in Houston or in Melbourne. So it's really trying to learn from best practice of other domains, but clearly there's a need to customize it to the unique requirements of a modified cell-based gene therapy.
Yeah, you covered that customization rather quickly, and I don't necessarily want to go into the, too deep into the mechanics of that, but I am curious about, you make a significant investment in manufacturing technology. You uncrate it, you begin to work with it, and shortly, in very little time, you have to turn around and go to the board and go to your investors and say, well, look, we've got some work to do here, right? Like we've got some tinkering to do. Walk us through, I guess, just the management challenge of that and how you kind of manage the company through that. I mean, there's also, managing a company through a whole bunch of unknowns with a significant investment.
Sure. So keeping in mind that what we began with was great academic science and what we now call an academic process, so a non-optimized process. And so in 2015-16, when we first got the company going, we had to make the decision, you could wait for years while you optimize all of this to get it, completely, what we now call our Plato platform, which is a commercial stage platform. However, who's going to fund you during that process? And do you really want to lose all of that time? So it's in all likelihood not even a feasible option. And so what we then had to do was to ask a very critical question at the beginning, which is which of these optimization steps do we absolutely need to do before we get into the clinic? And which can we do while we're treating our first patients? Because we had to get into the clinic to treat first patients to show that there's a positive signal, which then allows you to raise money and to fund and build the company. And so really there were only two things that absolutely had to be done right at the beginning. Is first, the quality of the stem cells had to be, absolutely optimized. And secondly, which some people just don't think of early enough, is you needed to cryopreserve the product, because that's a step that if you do after you've started your clinical trials, FDA, for example, would determine it as a different product, a frozen versus a non-frozen product. So those two steps had to be done early. And that's one of the benefits of bringing people like Kim Warren into the company, because, most people actually wouldn't know that as a very first step, and sort of the light bulb gradually goes on when it's too late. So I think that was absolutely essential to get that right. But then we could start our clinical trials and we could generate data, fund the company as we optimize the next steps. And there were really three major things that we then had to modify in the product. One we've talked about, which is automation. Mm-hmm. And, the nice thing, the reason why it was okay to have an iterative process is we were treating patients along the way with a manual process, with a non-automated process. So even though it took years to be able to develop the automation, refine the algorithms, have pre-IND meetings with FDA, reach agreement as to what data needed to be generated, generate the data, and get the regulatory clearance to pivot to automated, at least along the way we could treat patients. And so we could continue to build the company in parallel and then just show FDA what they call in vitro comparability. So an in vitro comparability process change to pivot manual to automated. Mm-hmm. And the exact same thought process applied to the vector. So we of course use these viruses, a viral vector that allows us to infect or to transduce the patient's own stem cells to deliver the healthy gene. Mm-hmm. And we started with an academic vector, the type of vector that's been, bouncing around NIH for years. And it's a good workhorse. It clearly works. And we showed that in our first 8 patients. But we wanted to optimize it. We wanted a really state-of-the-art vector. So the exact same dynamics. Start with the academic vector, enter into discussions with FDA in the form of a pre-IND meeting, reach agreement as to what type of data needs to be generated to pivot to our state-of-the-art vector, and then incorporate that mid-trial. And then the final was very similar with our approach to conditioning. So it's been this, the old analogy of working on the airplane as you're flying it. We out of necessity had to start the trials before the process was fully optimized. And then it just becomes a discussion with the regulators as to how do you pivot to these, how do you incorporate these upgrades, and what type of data do they need to see in order to approve these modifications.
The Business of Biotech is brought to you in partnership with Cytiva. Together, we're committed to helping the leaders of new and emerging biopharma companies navigate the financial, organizational, human resources, and regulatory waters you'll encounter on your way from discovery to the clinic and beyond. Check out a host of useful resources for biotech leaders at Cytiva's Emerging Biotech Accelerator at cytivalifesciences.com/emerging biotech. That's CYTIVA lifesciences.com/emerging biotech. How much of the work along the way has been, completely internal versus leaning into vendors, suppliers, CMOs, and what's that been like if you are relying on those outside folks? What's it been like kind of pulling them along?
Yeah, so there's about 130 employees at Avro, and I think if we ever calculated the number of FTEs working with our partner companies, for just the numerous things, CMOs and CROs and, critical raw material producers, it's many times that. And so, I think the conventional way of thinking, even in gene therapy, was that you wanted to insource everything and that you wanted to build these big bricks and mortar facilities and own each piece of the value chain. And of course, there's a lot of challenges with that. One is it's incredibly expensive, and you need to do that early when the cost of capital is enormous. But number two, it's inflexible, and it doesn't lend itself to globalization or changing demands of volume. So we took a very different approach as we mapped out that value chain, and we asked ourselves, which elements of it do we want to own, control ourselves, and which elements do we want to rely on partners? And the parts that we figured we had to be the best in the world at is process development, to continue to refine and optimize the process, and really automation and large scale vector process development production. Everything else, we wanted to just figure out who are the best partners to work with. And so, to sort of go right to the end, with us owning the component where we have fully automated manufacturing, we can just pick up one of these automated pods about the size of a dishwasher and just plop it down in a low-grade clean room in a CMO anywhere in the world. So we need a CMO partner. And for that, we've established multiple. We have manufacturing partners in the US, Australia, Asia, and Europe. But even still, even though we rely on CMOs, the critical element, which is the automated manufacturing, is still ours. And I think that at this point in time in cell and gene therapy, because the field is absolutely exploding, I think there are 600 or 700 clinical trials going on across almost you name it, pick a body part head to toe, gene therapy is being studied. It creates leverage for, not the person writing the check, but the person receiving the check, the CMO or the CRO. And so actually there's a real finesse and a skill set to be able to, for us to be a good partner and to make sure that we build up the skills to really interface with the CMOs and CROs in a highly efficient manner, because their businesses are all booming. Yeah.
So I just want to make sure I understand that correctly. You're saying that it's sort of incumbent on you to make sure that you're going to get that real estate, that IP. Is that what it comes down?
Yeah. Well, just that there's such a demand for, say, clean room suites in a CMO that you better be a good partner. And if you want to get to the top of the list, the way to be a good partner is to be efficient, to meet your timelines and to have highly skilled people to interact with their people, as well as to help with the training. So it's maybe one day the leverage will swing in the other direction and the manufacturers will have all the leverage. But I think right now there's more demand than there is space in CMOs and CROs. So it really behooves you to figure out how to partner efficiently with them.
Yeah. So you've talked about you just mentioned the fact that the space is exploding, the market dynamics. What's your assessment of where we are in terms of, our ability to leverage cell and gene therapies to address, you stick with the indications that AVROBIO is working on, but versus the standards of care, like where are we in this still relatively new space?
Yeah, well, so first to answer broadly about the space, and then Avro specifically, as I mentioned, that there are 600 or 700 clinical trials going on right now, mm-hmm. and I think 40 to 50 of them are phase 3, meaning registration trials, getting near the finish line, and there are 5 or 6 already approved gene therapies on the market. So if you just play this out, even a few short years, if normal attrition rates, clinical development attrition rates apply, excuse me, meaning the same attrition rates that apply to small molecules or biologics, mm-hmm. roughly a third of them will get to the market. And so what that means is that dozens, and a few years after that, hundreds of gene therapies should be arriving on the market. And so that will just mean a complete rebuild, transformation of the healthcare system. So this hopefully will transform the lives of patients, their families suffering from genetic disease. But even the delivery of healthcare has to change. The way healthcare is reimbursed is going to have to change. So just a dramatic difference from anything from cancer to rare disease to cardiovascular disease. So really, it's an incredible point in time. AVROBIO ourselves have targeted one piece of this. And our whole focus is in something called lysosomal storage disorders. And many people know of these diseases because they were the foundations for the building of companies like Genzyme and Shire, where mm-hmm. they developed enzyme replacement therapy. Lysosomal storage disorders are essentially a monogenic disease where an enzyme, or often an active protein and oftentimes an enzyme, is not produced. And the lysosome is the cell's recycling system. So as a result of this enzyme being deficient, eventually fatty substance or toxins accumulate in the cells, and it just results in a number of debilitating effects. Often life is cut short, and the life that these patients live is debilitating. So I mentioned companies like Shire and Genzyme pioneered this field 20 years ago. The whole approach was these patients are missing an enzyme. We'll give biweekly infusions of enzyme that take about 1 to 2 hours. The patients are asked to go to a hospital, sit in a chair, and get a 1 to 2 hour infusion of enzyme every 2 weeks. Mm-hmm. And that was a godsend. However, the big deficiency in that approach relates to pharmacokinetics. And what that means is that if you test the patient's blood 12 hours after one of those infusions, the enzyme's gone. So you have this big bolus of enzyme gone 12 hours later, and then none in the blood for almost 2 weeks, and then they get another infusion. So the whole premise of gene therapy is, and the reason we formed Avro, is what if you could deliver 24/7 expression of enzyme by having the patient's own stem cells and the progeny of their stem cells not only manufacture protein, but distribute protein head to toe. Mm-hmm. And so, in its most simple way of describing it, it's a better pharmacokinetic distribution of enzyme, and hopefully this steady, bathing cells, tissues, organs, and enzyme 24/7 should result in better outcome. And that's what we're hoping to demonstrate. We're 12 patients into this journey, so we still have a lot to demonstrate, but we're running 4 separate clinical programs across 4 separate lysosomal disorders. Mm-hmm. And we hope to be able to demonstrate not only that we can untether these patients from the biweekly infusions, free them up from, half a day every 2 weeks of their life, but more importantly, halt disease progression instead of just slowing it down, which is what today's standard of care is able to accomplish.
Yeah. So you mentioned four clinical programs. Your pipeline's a bit deeper than that, though. What you've got 6 or 8 candidates in the pipeline right now?
So we have 4 really sister indications, all lysosomal disease: Fabry, Gaucher, Pompe, and cystinosis. And we haven't as yet undisclosed pipeline behind it. Mm-hmm. But what we've tried to build as a company is a very congruent set of diseases with comparable biology. And the majority of these lysosomal disorders are very well understood. The nice thing about them, and this is because there's been 40 years of research and 20 years of clinical development behind them, is that due to this missing gene, there's an insufficient amount of enzyme, which leads to an accumulation of substrate, which leads to all kinds of disease symptoms. Mm-hmm. And so it's a very well understood path. So by putting the enzyme back into the story, it just clearly is able to do what the cells weren't able to do before, which is to clear out the substrate. So we've demonstrated that so far, early days, but in 9 patients across 2 trials in Fabry disease. And we do believe that having established proof of concept in one of these lysosomal disorders certainly should in some way change the probability of success in the sister indications, because the approach is very comparable. And because these are so related, we're able to develop this one, what we call our engine, which is our Plato platform. So the lentiviral engine. And it's almost like each of these diseases are like an SKU of each other, where it's the same cells, the same vector. We just switch out one disease-specific transgene, and we pivot from Fabry disease to Gaucher disease, for example, or Gaucher to cystinosis. So it allows a much more cost-effective development because we're leveraging our Plato platform really across each of these. Yeah.
I'm curious. You talk about you've mentioned a couple of times like the success ratio. You talked earlier about the ratio of cell and gene therapies that are making it beyond the clinic. That's higher than typical pharma, higher than really most biopharmaceuticals. To what do you attribute that? What's the what is it about this science that contributes to a higher potential success ratio?
Well, I think that you're directly addressing the root cause. You're not trying to address symptoms. I mean, small molecules are like molecular saboteurs that can gum up the works or block or stimulate. Whereas here you have a missing gene, and mm-hmm. as a result of that missing gene, you can't produce a protein. Here we're putting the missing gene back in play. And so you can now produce the protein. So I just think it's addressing the problem at a deeper root cause than traditional drugs.
Yeah.
So I would like to say though that big picture, I absolutely agree with you, and I'm just incredibly bullish on this field. I think that there still are some kinks to work out between gene therapy where it is today and truly becoming that first line therapy across the board. And if you think of what's transpired just in 2020, I think that it highlights that, this is hard work and there are some developmental challenges. I'd highlight in the AAV type of gene therapy, which is different than what we do, but for AAV gene therapy, there have been some unexpected safety and some unexpected durability signals that have popped up this year. And, mm-hmm. Most of the world is focused on the safety issues due to the tragic deaths of some patients in an Adentes trial recently. And they seem to be liver toxicity and immunogenicity related. And it appears to be an emerging trend because we've seen clinical holds from companies like Solid and Pfizer going all the way back to Dimension. And it's even been seen in Jim Wilson's large animal primate data. So it's unclear whether this generation of AAV or whether they'll have to wait for a next generation to address this. And is it dose related? Is it something that can be mitigated by the dose? But these are the types of things that are now being focused on. And then secondly, the world was really surprised by FDA's reaction to BioMarin data a few weeks ago, where they decided to ask for longer term follow-up because you could see a diminution of effect of this AAV gene therapy over time. So those are two issues which I think on the AAV side of the ledger are occupying an awful lot of attention and focus right now. And as I said, it will either be addressed by this generation. Already there are companies popping up that are designed to address this in the next wave. And then in my world, in the lentiviral gene therapy world, the historic challenge was the tolerability, not of the gene therapy, but of the conditioning agent. Is a few days prior to receiving a lentiviral gene therapy, the patients receive a dose of conditioning, which creates some space in the bone marrow to allow these stem cells to migrate and engraft. And the historic regimens were not very well tolerated. They were they had toxicity associated with them. And gradually, the research and approaches are refining towards a more gentle approach to conditioning. So it's these issues that are occupying a lot of attention in gene therapy right now. Big picture, it really seems to be working. It's incredibly exciting. Healthcare should be changing. And now it's just how do we address some of the issues to truly make it a safe, effective, well-tolerated, and single-dose therapy.
Yeah, excellent. It's exciting stuff. What looking at AVROBIO, what's the next big step for you?
So as I said, we've dosed 12 patients now, and I think we're seeing a consistent signal. And in normal world, 12 sounds like a very small number, but in gene therapy, because the effect is so profound, that registration trials could be as few as 20 or 30 patients. And so our thoughts are moving away from getting into the clinic and showing proof of concept to getting out of the clinic and starting to make those long lead time investments to build a commercially approvable product. Mm-hmm. So the type of skill sets in a company to commercialize is very, very different than just the early science and early clinical skill sets to show that the gene therapy is working at all. So we've recently built out a commercial team, and we brought in someone who led this function at Genzyme. We built out a reimbursement team, someone that's led the function at Avexis, one of the few companies to have achieved reimbursement in this field. And also outcomes research, also a real leader in the field from Avexis. So we're starting to change the phenotype of Avro to be a company that is not just proving ourselves that this works and optimizing our Plato platform, but really a company that's trying to make those long-term investments to prepare ourselves to, get our positive BLA approval and obtain reimbursement, value-based reimbursement, and also just some of the CMC issues to be able to really mass produce. So a lot of those are very long lead time items. And so we're making those investments now.
Yeah, well, that's exciting times. And I know the investment community likes to see that kind of activity in a company, hiring CMC professionals and chief medical officers and that kind of thing. So, congratulations on the success.
Thank you.
Yeah, we're actually short on time. Any concluding thoughts you wanted to share with us, Geoff?
Well, I just to say that when I got into the field of cell and gene therapy, I think you could fit the whole field in a small room. And some of the early congresses almost felt more like self-help groups where we're trying to encourage ourselves to continue because the challenges seemed formidable. There were unaddressed safety issues. The investment community had abandoned us. And it just felt like, that this may never happen. And now if you advance the clock to 2020, for example, at an ASGCT meeting or one of the main congresses, they take up an entire convention hall. And so part of the reason that we're going to be successful is just the amount of energy units that are now poured into this field. That there's big pharma is completely invested in this, biotech is invested in it, the investment community, but now tens of thousands of brilliant scientists. And so some of the issues between, the field getting forward there's just such a raw critical mass of talent and resources that it really does feel as though, in a few short years, this will be mainstream medicine.
Yeah. Very cool. I, for one, Geoff, am thankful that you did not pursue the Kraft mac and cheese path.
Yeah, well, I still eat Kraft mac and cheese, but I think I'm glad my career went another direction. Thank you.
Yeah, as are we. Thank you for joining us.
All right, take care.
That's Geoff MacKay. I'm Matt Pillar, and this is the Business of Biotech. We're produced by Bioprocess Online in partnership with Cytiva, which offers a trove of great resources for emerging biotechs at cytivalifesciences.com/emerging biotech. That's CYTIVA lifesciences.com/emerging biotech. Check that out and go to bioprocessonline.com right now to sign up for our newsletter, which is full of valuable content created by and about your biopharma peers. In the meantime, thank you for listening.